ist alles prima.
Okay.
Soll ich noch schnell 2, 3 Stühle da hinten reinquetschen? Also Stühle habe ich. Ich muss gerade noch mal. Ja, wie du machst.
Hello, everybody. We are ready.
The online participants are all ready.
We have lots of patience, so no problem. Waiting for everybody.
May I ask the project presenters, session 3 to come here? Okay, everybody. The project presenters of session 3, I would like to ask to join at the front. So if I'm not sure who's presenting food diverse, whole food, mild fruit. Could you please come to the front that we save time? Me wine. I think it's online.
Yeah.
Trust Farm. It's also online. Climaqua. Perfect. MCM Forest B. Okay. Oleoform. Oleoform is online. Cell for Chem. Synbiomed. Apple Biome. Impress. If. AgroBioConnect and Clear. Okay, Clear is okay. Great, thank you. Perfect. Sorry for the inconvenience. We need to have the wall closed. The technicians are already gone. So all remaining seats can be taken if you like, or you can also move a bit the chairs if you like. And then sit. Otherwise, I hope that we will be finished in about half an hour. So if you can stand that time, then if you're okay with that. Sorry for the inconvenience. Okay, we will start with the same setup for the session 3. Again, the bunch of pitch talks now from the SARS-CoV-2 Core Organic Call again, VOS Call 1, Oracle Biotech projects, and SASCROP-FATSCHE projects on agrobiodiversity. I will share the screen with the online participants. Okay. Petra or Ivana, I can see you. Can you say something if you can hear us? Okay, great, thumbs up. And can you see the screen?
Yes, we can.
Yes, we can.
Wonderful. So, okay, again, 2 minutes per project. And for the online participants, I will again move my mouse over the screen. Then I would like to ask, yeah. No, here we go. Okay.
Yes, thank you. Welcome back. I'm Van Waan, I'm coordinator of the Foodiverse project, and we're moving a little bit away from the more technological, because our project was interdisciplinary, but with a strong focus on the social sciences. We had agronomists and nutritionists, but most of us had been from geography, sociology, and more from the social sciences. And you have talked in one of the sessions about agrobiodiversity, and that was also one of our starting points, thinking about food systems that should be diverse. But we didn't want to stop at the farm gates, but we said that we need to recognize diversity in the entire food system. So that's what we did in the project. We took a food systems approach, and we didn't start at the production, which you would normally do, but we flipped the food chain and we said we need to start by the consumer side, and we need to investigate diversity in food cultures. And then going backwards along the food supply chain, we did a market audit. We audited over 2,000 products in 5 different countries. And towards the end, we also included food governance, because we thought we also need to recognize diversity in governance. And it was very interesting to see how, for example, European policies are implemented at national levels. And we did all these 3 research packages on food culture, food supply, and food governance in 5 different European countries. That is Norway, Poland, Germany, the UK, and Italy. And we did in all countries the same approach. And that helped us to distinguish between the different dimensions of diversity. And as you can see, there are different kinds of diversity. And that led to the second part of the project, where we set up living labs, which is a little bit fashionable at the moment to say we worked with existing initiatives, and these initiatives helped us shaping our research questions. So it was a little bit what we have heard this morning in the pitch. It was a little bit an iterative process. And from all these results, we came up with a diversification roadmap. And if you would like to know more about our diversification roadmap, you will have to come to the next session in Berlin, in this room, I think.
In this room, yeah.
Thanks a lot.
Thank you. Perfect. Next one. So.
Thank you. Good afternoon, everybody. I'm Riccardo Dulli. I'm a research fellow at the University of Padua in Italy. And now I'd like to briefly introduce you to HO Food. HO Food is an international project focused on the development of a novel high-pressure technology to increase the preservation of ready-to-eat and fresh-cut organic food products, while maintaining their high nutritional value and consumer attractiveness. As you all know, fresh-cut organic products are well known for its freshness and quality, but they often face some challenges, especially regarding the short shelf life and the higher risk of pathogenic contamination. The current technologies are limited in enhancing the preservation of these products, and this is strongly connected to an alarming statistics, which is that 1/3 of the world's food production goes to waste. So our approach is to use a low temperature CO2-based product process in order to inactivate microorganisms present on the surface of our products and to increase the shelf life without compromising the nutritional quality of the products and the consumer attractiveness. So in our case studies, we demonstrated how the process was effective in enhancing the shelf life of different products. And so I will show you more results later. So if you're interested in knowing more about the HFood project, I will invite you to stay here in this room, so it's easy to dive into the science behind our approach and to know more about our findings and our next steps. Thank you.
Thank you. Okay. So next, HFood Core Organic project.
Okay, good afternoon to everyone. And our project was born from the idea that probably when we consider organic products, we consider them like old-fashioned ones that we have to produce like in the past. But in our opinion, this is completely wrong. If we think to preserves or dehydrated fruit, we can have with conventional processing a detrimental effect in terms of nutritional characteristics, sensorial properties, and other kind of aspect. Also strictly related to the sustainability. So with this project, we try to apply emerging non-thermal technology involving experts in food processing, but also in consumer science and sustainability aspects. So our assumption is that we have raw materials with very high nutritional quality, no pesticide and so on, as you know, but often they are more perishable, and we have just few products in the market, like conventional ones. We need technology, we need innovation in technology in order to better preserve these characteristics. So we try to obtain snacks, minimally processed fruit and vegetable with some functionalization using pressure, electric fields, and other kind of physical principle. But also we try to valorize byproducts, because we have added value byproducts from organic fruit and vegetables because we don't pesticide, so we can use it to produce ingredients and to have a more affordable approach. And of course, we have to consider focus group with consumer and also aspect related to to LCA. Thank you.
Thank you. Perfect. Okay, Meewine, I think it's online. So can you switch on your
Can you hear?
Ariana? Yeah, we can hear you.
Oh, perfect. Thank you. Welcome all. Good evening. Here is a pitch of the Meewine project, which introduced an innovative treatment for the stabilization of wine with some potentialities for industrial transfer. The Meewine project aims at supporting the production of quality wines and to deal with the issue of the wine instability, especially protein stability, which is a major concern in the production of white wine. Next, okay. The bottled wine needs an efficient stabilization in order to withstand extreme conditions during the storage and in order to meet the preferences of the market in terms of quality. The solutions which are currently available for the industrial stabilization of the wines present several disadvantages in terms of economic impact, which relate to the efficiency and to the cost efficiency of the process. In terms of environmental impact, which is mainly associated to the energetic cost and to the amount and nature of the waste that are produced, and can affect the consumer's confidence reported as the main social impact. Next, please. So to overcome this limitation and these disadvantages, the Meewine proposes a combined approach, which involves the introduction of a high-performance and a nano-engineered material and a technological approach that is inspired to the circular economy. So recycling, reusing, and so on. The result is an innovative and mild technology that boosts innovation for a competitive and global wine chain, and obviously to produce higher and higher quality wines. We'd like to thank you for your attention, and more details about the project outcomes in the Berlin One room.
Thank you. Okay, so we switch now to Foscol One projects. Yeah.
Good afternoon, all together. Risk, climate resilient, and responsible innovations in potato. It's all about potato and adaptation to climate change. The problem we all know, climate change threatens global potato production a lot, especially in Africa. We have 2 African partners. Impacts on yield, quality, income, and food security are obvious. Smallholder farmers particularly are affected, like with so many other crops. What did we do and what are we presenting later? Our solution is, first of all, a comprehensive sector analysis we did in 2 African countries and 3 European countries. Breeding for Resilience was one work package. It was a combination between Irish and Kenyan partners, economic innovations and responsible innovations, where the social scientists from Wageningen University came in and looked to the technical stuff, if they're doing it correct, thinking about the end users, next users, and if the innovations will have an impact. Yeah, so expected impact or impacts would be basically 4. I'm presenting a bit more on that. Comprehensive climate smart potato toolbox. It took us a while to understand what we mean with that. Then the second one, climate resilient varieties with market desired traits. Third one, enhanced productivity and reduced losses. It was also a bit about post-harvest losses in the chain, and inclusive stakeholder-driven solutions was the fourth one. Yeah, and we engaged with our research team, adopt climate smart practices with a lot of stakeholders and partners. And if you want to join me later, you have to cross the new Berlin Wall in Berlin too. Welcome.
Yeah. I hope it doesn't stay that. Okay. Okay, Trust Farm. I think it's an online presentation by Osama Ahmed. Can you hear us? Can you switch on your microphone, please? Petra, can you check if
I'm already checking if
Osama
Ahmed is there, but frankly spoken, I can't see a name like this.
I think I saw him in another room, but okay, unfortunately he's not here. Hopefully he's then in Berlin too. Okay, then we need to move on to Climaqua. Okay.
Yeah.
Yes. Thank you.
Yeah. Hello, everyone. So in Climaqua, we try to make the world a better place, as all of you, I guess. But what we found out, it isn't as easy as it seems. No, seriously spoken. In Climaqua, we aim for a disconnection of aquaculture and agriculture. So why do we want to do this? Aquaculture takes a lot of the agricultural products for feed production for whatever species is grown. And aquaculture is also the place where a lot of residues appearing, side streams or whatever. So we have actually the option to convert side streams from aquaculture directly into new feed and then feed it to whatever species. I have been doing this for a couple of years already, but I never asked myself, does it actually make sense from an economic point of view and an environmental point of view? There are different ways to cultivate microalgae, heterotrophically and autotrophically. Microalgae is the food for our fish, for instance. We aim in Climaqua to find out what process should be applied where and what environmental conditions actually benefit a certain process. So luckily, the FOSC gave us the chance to work with partners from the north and partners from the south. So we could compare different climate conditions and then correlate it to our process performance. So if you want to learn more about growing microalgae and where it should be done, then visit me in room number 32.
Thank you.
Thank you very much.
Okay. Okay, I think actually, okay, I think feeling actually was cancelled. Let me just think. Feeling. Yeah, I think that presentation was cancelled. That's yeah. Sorry, change in program. So we need to move already to Paris. To the Araco-Biotech presentations. So the first one would be MCM4SB.
Yes, the change here.
Yes, or the arrows.
Okay, hi everyone. My name is Trygve. I'm from Norway and I am the project leader of this MCM4SB projects. So a few words about why, what, and how. So first, industrial biotechnology, which is the sector we work in. It develop and it use microorganisms into biochemical cell factories for sustainable production of a wide range of different value-added compounds and chemicals. And of course, a lot of the research is on the products, which products to produce and how much you can produce. But also now there is a growing demand for replacing today's sugar-based substrates, that is the carbon source that you feed the bacteria with non-food and renewable alternatives. And this is partly our focus here. And one carbon C1 substrates, they can represent such alternatives. And in our project, we genetically engineer metallotrophic bacteria for conversion of such C1 substrates, which in our case is methanol, methylamine, and carbon dioxide, into value-added compounds. Now, metallotrophic bacteria, these are bacteria that are specialized in the sense that they have the inherited capability of using these compounds. So that is why we use them. And our project is a good mix of basic and applied science. I would say that when it comes to scientific challenges, there are, as always, very many. But in particular, when you work with metallotrophic bacteria, they are much less characterized than the traditional organisms that are used in industrial biotechnology. It comes to our understanding and knowledge about the biochemistry, the genetics, physiology, and also when it comes to genetic toolbox that you can use to do the genetic engineering. This is a simple illustration of our project. I will tell much more about that if you come to, where do they come to? Paris? Paris. If you come to Paris. Yes, thank you.
Okay, thank you. Next biotech project is Oleoform. I think it's online by Cristina Gonzalez Fernandez. Can you switch on your microphone, Cristina?
There is a Christina on the participant list. She's trying.
Okay.
Christina, you find at the bottom, you find at least 4 buttons, and the left one is unmute. I tried to ask you now. Okay, so if you click to the arrow next to it, It should show you a list of activated microphones. Or of the non-activated. Probably you need to activate the proper one.
Otherwise, I suggest that we move on and maybe, Christina, if you try to fix this in the meanwhile, then we might miss your pitch talk, but hopefully not the presentation.
But maybe we can see if Christina can please switch on her video so we can understand what is the situation like. Christina? As Stephanie has proposed, and also as was explained, you need to connect microphone and your speaking device to headsets, or there is a long list. So try to do the correct connection and have the presentation then properly. And we are here meanwhile in the chat, we can help you while other pitches go on. So please just write what is the situation.
Okay, I think we move on because we are short on time then. Okay, thank you. So then it would be Cell for Chem.
Yes, it's me. So my name is Heike Streuber and I'm coordinating the Cell for Chem project. It's about engineering microbial communities for the conversion of lignocellulose into medium chain carboxylates. So actually, medium chain carboxylates are chemicals that can be used in many different industrial branches. But at the moment, they are still from palm kernel oil or fossil resources. But there's a more sustainable alternative, and this is producing them on the carboxylate platform at biogas plants. And this works because the carboxylate platform and the biogas process, they are quite similar to each other and have many things in common. So the biogas plants, many different substrates can be used as substrate for producing biogas, but only few of them are applicable also for the carboxylate platform. And therefore, in our project, we use lignocellulosic substrates because the aim to expand the applicability of the carboxylate platform, but there are many different bottlenecks that we have to overcome. And we tackle this challenge on 3 different levels or on 3 different fields. There we have the genetically modified bacteria that work for us. We construct synthetic consortia of bacteria specialists, and finally we also optimize the technical process. And if you're interested in the results of our project, and if you want to know what role this little creature plays in our project, then come to Paris and join us. Thank you.
Okay. Last presentation. Okay, it's going to be on me.
So, hi, I'm Nadine. I'm presenting the Synbiomed project today to you. I'm a PhD student working on this. And the problem we are facing in the Synbiomed project is actually that the amino acid methionine and its analog HMTP are still exclusively produced from fossil resources. And this is an exception among the protein organic amino acids. These compounds are mainly used as feed additives, and with a cumulated annual production volume of more than 1.5 million tons per year, there is a need for a more sustainable production process. However, at the moment, there is no fermentation process based on renewable resources available. So we proposed a 2-step process where in the first fermentative stage, an intermediate is formed, which is DHB, and we are actually using microorganisms for this, which use sugar as the feedstock. And in the second step, DHB is then chemically converted to HMTP with a near to 100% carbon yield. And this gives actually a rise to a more sustainable and cost-efficient production. So Symbiomed focuses on this first fermentative stage because getting access to DHP is actually not that easy. It's not a natural metabolite. So we are actually relying on the great potential of synthetic biology to produce this compound. And if you're interested how we are doing it and how we optimize DHP production to actually at some point be competitive to classical chemical synthesis, then I'd like to invite you to Room Paris, where you get to know all the things. Thank you.
Okay, from Aeroco Biotech now to agrobiodiversity projects, and I invite Apple Bio. Here, just on this.
So good afternoon. My name is Maria Jose Arantana, and this is not a pitch talk, it's a Pixar talk. And where biodiversity is on danger and Apple Biome goes to the rescue. So actually, Apple is a clear example of intensive management practices, which together with the monovarietal cultivation of the crop, reduces a lot the biodiversity in the field. Also, it reduces the ability of the crop to fight against new challenges, okay? So to solve this project, Apple Biome wants to analyze the apple refug collection, that it's a collection of apple germplasm that has about 500 genotypes, that we are going to look at the microbiome in the leaf for study the diversity, how the environment, the management, and the conditions modify this diversity. So the benefit for that is for the agriculture, the breeders, and the society in general. So here you can see the partners of the project. And if you want to see more or see more Pixar pictures, you can come to see me at Kyoto. Thank you.
Thank you. Very creative. Okay. Impress us, please.
Okay. Well, hello everybody. I'm Cassandra from the Swedish Swedish Agricultural University of Uppsala, and yeah, our project is about biodiversity that provides biological pest control, and then I do not mean supplementing these animals to fields, but rather to capitalize on this natural asset that we have in our landscapes and our fields that provide pest control services to our crops. And we know that these animals are heavily affected by the surrounding landscape and by the management practices of farmers, as well as the changes in the climate. And what we really want to know is how we can make these predator communities resilient to changes in the future, which is especially important if we want to move towards more sustainable agriculture, where we rely less on chemical pest control. And so far in the studies and literature, it's not really measured how all these different aspects, such as landscape structure and management and climate, affects the functional resilience of these predator communities. So in order to do that, we started Impress project, where we have various partners and collaborators that work both in the social and the ecological sciences, that look at several questions regarding resilience of these predator communities. And in the end, we want to synthesize this data to give recommendations for the future. And how exactly we measure resilience in the field, you will just have to find out by coming to our presentation. So thanks.
Thank you. All right. So then go to Kyoto if you want to learn more of pest control, as well as
Stephanie?
now for yeah.
Yes, you need to stay there just to click on the slides for this presentation.
Yes. Okay.
Yes. So
the voice from the background.
yes. So AgroBioConnect, I think the title of the project, which is Connections in the Landscape and the Role of Landscape Complexity in Agroecosystem Sustainability, describes a little bit what we are dealing with, and it's a little bit connected to Impress, but we do look at the biodiversity from another point of view. Next slide, please. So we see that the main objective was to investigate the effect of the diversity of landscape under on the above and below ground ecosystem services. You see on the left the slides, the photos with different kinds of landscape. Next slide, and then this through the satellite images can be, let's say, reconstructed in a different way. And the next slide, you see what kind of information we can get also from the satellite images and analysis. So this is the phenology, what we call during the whole year about what is going on in the specific area that we are analyzing. Next slide. So what we do besides the satellite, we also check what is happening on the ground. I mean, on the ground in the sites where we are studying, and we have 6 partners working on that. We are in Poland, but we have from Denmark, we have from UK, we have from Finland as well, and Italy. And so we use common methods for trapping the above ground biodiversity, because we are focusing on both, let's say, the beneficials and the pests. Next slide. But we are also using new techniques. This is a new machine which is using, say, is capturing the vibration of the wings of the insects and then defining which kind of insects we are measuring. And for this reason, in the next slide, you see that we have also were able to focusing on very specific species of pollinators. This was found in the UK, and a new species, I believe, that can be seen even from the satellite. But we just also go on the below ground biodiversity, and you see we are using metabarcoding, analyzing both fungi and bacteria, and we are trying also to verify the nematodes. So whether there is an impact of the landscape on the below ground biodiversity. The benefits, it's knowledge for, you need to see, yes, that's fine. Go ahead, yes. On the Kyoto, you will have a possibility to learn a little bit more. I will not be there, but will be Gerard that will tell you a lot more. Thank you.
Thank you. Okay, and last but not least, Clear. Also, agrobiodiversity project in Kyoto. To use.
Yeah. So I'm Carla. I'm a PhD in the CLEAR project, and I'm gonna try to be very brief. So starting with our problem, in the last decades or several decades, agriculture in the UK and Europe has prioritized high yields or production, and often with a farm-centered approach, which on the one hand has helped to meet the demand, which is very good, but it also simplified our landscapes, caused biodiversity loss, and even more critically reduced the resilience we have for our landscapes. So in the CLEAR project, we believe there is a better way to do that and move from this simple or simplified systems to a multifunctional landscape. And the key here for our project is collaboration. So we want to bring different stakeholders together and discuss how to make a plan for a landscape in the different case study regions we're working, so that we can have more multifunctionality. And so how are we going to do that? We're combining socioeconomic as well as environmental data to understand what is the current state of the agri ecosystems we're looking at. At the same time, we want to understand what are successful sustainable practices that they are already implementing in the case study regions, and also what are preferences for shifts. So when we have this knowledge, we can start thinking about how to upscale the successful practices that are being implemented, as well as develop with the stakeholders some new methods of collaborative planning. So who benefits from that? First and foremost, the stakeholders and farmers, but we want to have generalizable results so that we can inform a shift towards more multifunctional landscapes across Europe. So, Mrs. and Kyoto.
Thank you. Great. Thanks, everybody. So a few.
Osama has found us.
Okay.
He's not ready to switch now. He was waiting in your.
Yeah.
So you should not divide cities.
Yeah, yeah. Okay. I'm just give me a second.
Yes, trust farm.
Trust fund. Here we go. Yeah. So Osama, can you switch on your microphone, please?
Now?
Yeah.
Do you hear me? Yeah.
Yeah,
Okay.
yeah.
Good. Good.
Yeah.
So,
Great.
sorry, I confused. I went to Berlin too. So, sorry for this. So, my name is Osama Ahmed. I'm the coordinator of the project towards resilient and sustainable integrated agro-ecosystems through appropriate climate-smart farming practices, Trust Farm. To next slide, please. Yes. So, our main goal is to design integrated agro-ecosystem. So to do so, we have 8 interlinked work packages. Firstly, the first work package was focusing on understanding the current context in the case study regions. Well, we have 5 case study regions. They are in Egypt, Morocco, Senegal, France, and Italy. And then we have established stakeholders platform to link directly with the platform, with the stakeholders in this in the case study regions, and to involve them to select the appropriate and best fit climate smart activities or practices. And then we have designed it together the best integrated agro ecosystem in the 5 case studies. I will give more information about our activities in the main session. Also, we have a work package for dissemination. We have several PhD and master students involved in the project. And also we have established or created Facebook and Twitter to disseminate the project activities. And also we have several publications in open access. Yeah. So this is our main work package and main activities. I will give more information in the main session. Thank you very much.
Thank you. And it's in Berlin too.
Yes, exactly.
Thank you,
Yes.
Matt. Okay. Just for a second. Blah, 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 blah. So we now go again to the breakout sessions. Before you go, is someone missing a mobile phone? I'm hiding it. No, it has a. Maybe outside? Okay, we will check. So, okay, please go to the breakout sessions. And then, yeah, we will come back together to a short wrap up and then we have a nice dinner.